I'm good? Yeah, there we go. All right, hello everyone. I'm Jeff. This is Nate, Sandy, and Danielle over there. And we're going to tell you about our population study of the eastern Massasauga rattlesnake here at Pierce Cedar Creek.
So, eastern Massasauga rattlesnakes are present throughout much of the Great Lakes region. You can see from this range map here in the upper right corner. They extend down into Missouri and Iowa, all the way up into Ontario, with 2 known remaining populations in the state of New York. Massasaugas prefer wetland habitat, however they will utilize adjacent upland habitat if it's available, such as here at Pierce Cedar Creek. And they are the only venomous species of snake in Michigan. Also, they're important ecosystems as both predators and prey, especially in controlling small mammal populations, because that's their main source of prey.
So throughout their entire range, their populations have been declining. And due to this decline, they have been listed as a candidate species for federal listing under the Endangered Species Act. The conversion of wetlands into agricultural lands has proved to be the major reason for this decline. But also the degradation of their habitat from invasive species, as well as direct human persecution, has also proved costly. And even though Michigan is the last major stronghold for the population, 33% of the historical population of eastern Massasauga rattlesnakes has disappeared.
And so, Massasauga conservation, it's difficult, it takes a lot of time, it's a lot of hard work. Anthropogenic habitat alterations have left the remaining populations highly isolated, which can reduce gene flow, reduce adaptability, and increase mortality via edge effects. Conservation biologists have kind of uncertain about the viability of the remaining populations, and so just really why the work that we're doing is so important. Doing these long-term population studies is really the best way to learn a lot about these populations and how they function. So you can see, and these studies are really good for land managers because it gives them like lots of great data to work with, and it can show them like if a threat is occurring, it can allow them to act quicker rather than later to a problem.
So, Pierce Cedar Creek has a history of Massasauga research that started in 2004, both in 2004 and 2005, and then again in 2008 and 2009. There were telemetry studies done by master's students from Michigan State University. So basically, they actually implanted these snakes with transmitters and then would track them and take data on their habitat, as well as they did mark them with pit tags. And then starting in 2011 and 12, U-Man Lee and that picture from the Michigan Natural Features Inventory led some volunteer surveys, I think for about 2 weeks each summer. And so they ended up with a total of 35 captures of 32 different snakes over that time period. And then in 2013, I was out here with my research partner, Brooke Keel, and we did a market capture study for the entire summer as part of the URGE program. And we were able to capture 78 recaptures of 51 different individual snakes. And then last summer, Nate and Jeff were out here and they beat us and captured 56 individuals 85 different times. And what's really interesting is that we were able to capture snakes from as far back as 2008 already.
So visual surveys have made up the bulk of our captures this summer. And basically, we just wandered throughout a various unit and just searched the vegetation for rattlesnakes. Once a rattlesnake is found, we take various environmental data such as soil temperature, air temperature, as well as cloud cover. And then we also mark the point with a GPS and flag it with flagging tape. Then the snakes are taken back to the lab for further processing. We measured their body as well as their tail to get snout to vent length. We sex the snakes and weigh them. And we also describe their rattles. Each snake is given a pit tag with an individual ID. And we also paint their rattle with a unique pattern so they can be easily identified in the field. We take blood samples for future genetic analyses, as well as we swab the snakes for the presence of snake fungal disease, which is a big problem arising.
So just the results so far from our surveys that we've done, 73 total captures with 49 individuals. These numbers are incredible. since we've done this prior, and we've already beaten our what they did in 2013 with only a month and a half of work. So that's awesome. Lots of more recaptures too that we're going to see, which is a lot better for the statistics that we use to determine the different demographics about the population. And getting more females, but still a good mix of males to females, which is also really good for those statistics as well.
So this is a map of all the captures of Massasaugas on the property from 2011 through this year, up through last week. And just to orient you a little bit, you have the wet lab, yep, thanks, right there, and then Cedar Creek, put that up, and Brewster Lake up there in the corner. So the yellow outlines are roughly the areas that we survey, and then in the blue we have all of the past year's captures of Massasaugas, and then in the red we have this year. So mostly you can see they're kind of concentrated this year in these few areas, but a lot of times as the season progresses, we'll see them and move into these other areas, like more by the lake and across the creek.
This summer we've incorporated various trapping methods to supplement our visual surveys. We have 4 drift fences with 8 funnel traps, 28 carpet squares, and 28 cover boards placed throughout the wetlands.
All right, so here's a map of how we placed out our traps here. These lines with the squares are the funnel traps with the drift fences, and then the green squares are the carpet squares, and the red ones are the cover boards. And we kind of use some GIS software to determine where we've captured snakes in the past, and we use that to figure out where we should best put our traps to get the most captures. And you can see that we've placed them like this. And also, other things that come into consideration when placing these things is like how wet is the area. If it's going to be covered in water most of the summer, it's not going to be a good place to put it. Also, how much vegetation is there. But we put these fences in, and it was a lot of work, and the less vegetation, the better for putting them in. And so we kind of just set them up in an alternating pattern of carpet to coverboard, carpet, coverboard, and we tried to space them out good enough so that the traps wouldn't interfere with the other traps, because we don't want to have any bias, and just we're trying to see if one trap might be a better way to capture snakes than another trap. And so this is just some of the early results from our traps. 15 days of trapping so far. We actually did catch a rattlesnake at one of our funnel traps. It was awesome. We were kind of losing hope on that. It happened just recently, but we've also caught lots of garter snakes, as you can see here. I had one with 4 garter snakes in it, which is kind of exciting. And lots of toads, frogs, and meadow voles, and lots of crayfish in these traps also.
So the cover boards, we've got 1 eastern Massasauga, and then we've caught 2 brown snakes and 1 short-tailed shrew underneath those. The Massasauga was actually on top of the cover board. And then we got 4 brown snakes and 5 garter snakes with the carpet squares.
Okay, I'm Sandy Bricklock, and I'm actually a biology teacher, and I just joined this group last week. Sorry about that. And I'm really interested in the snake fungal disease. And so this was first identified here at Cedar Creek last year. And it was this is the actual, go back one more time, it's showing the fungal disease on the snake itself. So it's kind of a dermatitis, and it's caused by a fungus, Ophidiomyces, that is a keratin-loving fungus, and so it gets into the scales. The scales have keratin in them. And so this fungus was first identified in captive populations of snakes, and several years ago it started turning up in wild populations, identified in Michigan up at the Grayling site, in 2013, and then last year it was discovered here at Cedar Creek. And so when I was looking for a project, it was suggested that snake fungal disease, we'd like somebody to take a look at it. So what I'm doing is I'm doing some background research on when this disease first started being reported in different states, and I'm looking at trying to map the report dates to see if we can see what are the patterns in the spread of the fungus. There's not a lot of information out there, so it is pretty interesting. It has been confirmed in several other other states, but they're just getting pretty good at identifying this through a molecular biology. So when they take their biological information, their measurements and so forth, they're also taking samples, which are then taken in, and a PCR test is run on them to identify the presence of the ophidiomyces, which they're thinking that this is very important in causing the snake bone disease. Some problems with this, though, is there's still a lot of questions. Is this fungus actually completely responsible? It does seem to be causing mortality. A lot of times the fungus has been an opportunistic pathogen in that the snake is compromised, the immune system is compromised in some way. And so there's a lot of questions around that. Is it because of the loss of habitat and all of the other problems with the snakes that their immune system is compromised, and so the fungus is more important? But what I'm finding is there's just an awful lot of questions and not a lot of answers right now. There's questions about what are the reservoirs in the environment? What I'm reading seems to show that the fungus is out there. Are there other organisms that are spreading the fungus? So I would like to be able to kind of look for that. I haven't found a particular project or some methods that I can use for that yet. As I said, there is a new PCR test available, but it's rather expensive, and so I'm kind of looking into what can be done. And there's still a question is the fungus actually affecting the population sizes? And there's been some studies that have shown that in populations that are in decline, the fungus seems to be pretty important. So I've got a lot of possible questions to ask.
So for the rest of the summer, we'll be doing our market capture surveys through August 14th. And then since I'm a master's student, I'll be out here in 2016 and 2017 as well doing market capture surveys. Then we'll be analyzing our data using Program Mark to look at things such as the population size, survival rates, and the density of snakes here. And hopefully by 2017, I'll have enough data to look at population growth. And then also, we'll look at the trap data this summer and determine whether or not I'll want to be using cover boards and carpet sprays and joint fences in the future to keep sampling, or we'll just continue on with visual encounter surveys alone. Also, Sandy will be mapping the spread of snake fungal disease. And then in the